Thanks, Klaus. Okay, now I think I told you that we have buffers in order to stay in the program, and the reality is we have to kill the first buffer, which is okay. So for those who have to leave, that's a good time to leave, because there will be a very short break of about 10 seconds when we directly switch over to the next speaker, who was actually also a speaker of last year. Last year I was a bit nervous because it turned out that the keynote speaker last year actually did not show up, but it was just a matter of seconds where these problems could be solved. And I'm very glad to have Shane O'Mara again participating. Last year he has talked about his book, In Praise of Walking. That was a bestseller in various countries, I think. And I'm now very curious about the delta, what you're going to talk about today, based on, well, the state of the world that has changed, as we know. So Shane, the stage is yours.
Thank you, Martin. And it's hard to believe it's a year since we spoke together last. And I had hoped that 2022 would be a better year than 2021, but I think fate is determined to direct us in a different direction. Anyway, so I am going to share my screen and let's get rolling from there. So hopefully you can all see my screen in front of you.
Yes.
Yes. Okay. So I'm going to talk about what are my core research interests. My book on walking, which Martin very generously mentioned, kind of evolved out of my core research interests, which are to try and understand the brain systems that are involved in spatial navigation, so how we move around in the world, and the brain systems that are involved in memory. And it turns out, evolution is a wonderful thing, that the brain systems that support spatial navigation are also the same brain systems that support our ability to remember what has happened to us, and also our ability to imagine what we might want to do. So I'm going to talk a little bit about those for the next couple of minutes. So the first description of the amnesic syndrome was about 130, 140 years ago. Two neurologists, a British neurologist, Robert Lawson, described cases where what gives them character is the almost absolute loss of memory for recent events. And more famously, Sergei Korsakov noted an amnesic syndrome where, again, especially characteristic is a derangement of memory. And the German neuroanatomist Hans Gudden studied these cases in great detail and concluded that brain damage in these patients was centered around a series of structures known as the anterior thalamic nuclei. You have an example of a post-mortem specimen here on the left, where there's extensive damage around the midline of the brain. And here on the right, you have 7 Tesla imaging of the human anterior thalamic nuclei. And on the bottom right, you have the corresponding nuclei in the rat. So just for quick reference, you have 3 anterior thalamic nuclei, the anterior, medial, and dorsal. Now, these are very complicated in terms of their connectivity. We've done many studies in collaboration with my colleague, John Aggleton, where we've looked at the connections between the anterior thalamic nuclei and other brain regions. And I want to emphasize one particular set of connections, which is to this brain region, which I'm going to talk about now, the hippocampal formation, which is a large and intensively well-studied region of the brain. What has happened remarkably is that despite the clues given by Gudden, by Lawson, and by Korsakov, most memory research focuses actually on the medial temporal lobe, which is this lobe on the side of the brain, and the hippocampal formation, which sits within. So if you look at this video, you'll see the hippocampal formation on both sides of the brain spinning in a circle there. And if you go to the literature, what you'll find is that there are standard models of memory that focus especially on the hippocampus and how it talks to the neocortex, and the anterior thalamic nuclei are essentially ignored. And this is a strange lacuna in the literature, but it's one that's explainable, partly because of this chap. This is patient HM. HM underwent a surgical operation in the early 1950s, 1954, for epilepsy, which was refractory to drug treatment. In other words, it did not respond to drug treatment. And the consequence was that his epileptic seizures were abolished, but he suffered a severe and intractable amnesia. And this did not resolve over the subsequent 50 or so years of his life. And when his brain came to post-mortem, indeed, he had very, very discrete damage to the hippocampal formation in one or two adjacent areas. And subsequently, it has been found repeatedly that the primary site of damage in Alzheimer's disease, disease particularly associated with memory loss, is indeed the hippocampal formation. Moreover, HM's amnesia was not general. It was very, very specific. His short-term memory was normal, so he could repeat a telephone number. He could learn new motor skills, and his IQ remained unchanged. He could, for example, if you look to the top left here, learn how to mirror write, and he got better at doing this over the course of days. He became very good at card games where you have to change your strategy as the reward pattern changes. And many, many subsequent studies, probably in the thousands of studies now, have shown that lesions, that is damage to the hippocampal formation in rats, in monkeys, and in humans, causes a dense and non-resolving amnesia. One that, in humans at least, is accompanied by insight. HM says here that what worries him is that being in this condition is like waking from a dream. He doesn't know what happened just before. In other words, his life is a series of unconnected episodes. There's another reason why we're interested in the hippocampus, and this is the Nobel Prize winning work of John O'Keefe. Some 50 years ago, O'Keefe and his student John Ostrowski implanted electrodes in the hippocampal formation of the freely moving rat and recorded, using the primitive technology available at the time, action potentials, that is the discharge of individual neurons in the hippocampal formation. And they found something really, really astonishing, which is that the cells of the hippocampus don't care about what you are doing. They don't respond to rearing, to sniffing, to chewing, or any of those kinds of things. Instead, what they respond to is the position in space that you occupy. These cells have become known as place cells as a result, and are now regarded as the core of the cognitive map, or the GPS system that the brain engages in. I'll just give you an example of what such cells look and sound like. So just to show you what you're looking at here, this is a rat moving around with the LED lights on the recording implant, and he's navigating in a little box. Every so often you'll see a food pellet appear, and he runs and eats that. And most of the time you're going to hear very little, but just occasionally you're going to hear something. So listen out for that something. That is the discharge of a single hippocampal neuron. And these cells fire when the animal is in the upper right quadrant of the environment. Or this particular cell does. And it's silent when the animal is elsewhere in the environment. So really quite astonishing cells. In the interim, head direction cells have been discovered. These are cells that have a compass-like property, and grid cells, which also shared the Nobel Prize in 2014. And these are cells which provide a metric in space. Now, what we have done is to try and recenter the argument by focusing on the anterior thalamic nuclei. So in the series of experiments I'm going to show you now, what we've done is to lesion the anterior thalamic nuclei using a chemical lesion, a neurotoxin, deplete the number of cells in the anterior thalamic nuclei, and record it from the part of the hippocampal formation that speaks to or connects with the cortex and with the anterior thalamic nuclei. So on the left here, you can see a normal histological slide. On the right, you see the cells have been depleted extensively from the anteromedial, the anteroventral, and the anterodorsal nuclei, and the cell count goes down dramatically. The first thing to note is that animals' performance after lesion falls to chance on a standard alternation task, where they have to alternate where they go in space from one test to another. The chance is 50%. Sham controls are at about 80%, normal controls are at about 80%, but the lesioned animals are at 50%, given that it's a two-choice task, chance is 50%. And what do we find when we record cells in the subiculum, which is this area that the anterior thalamus connects with in the hippocampal formation? Well, what we find in the control animals is we get place cells, we get head direction cells, we get these amazing border cells, we get these fantastic looking grid cells, and we get occasional non-spatial cells. But in the lesioned animals, what we find is the spatial signal disappears entirely. In other words, the output of the hippocampal formation depends on an input from a separate anatomical structure, which is really quite remarkable. Moreover, when we do reversible lesions, what we find is behavior when the animal is under the reversible lesion, which is the local anesthetic infusion into the anterior thalamic nucleus, their behavior falls to chance while they're under the reversible lesion. But when the lesion and the neuronal responsivity drops, as well as spatial selectivity drops away. But it turns out to be the case, remarkably, that as the mucimol, which is the local anesthetic, is metabolized, the responsivity of the cells comes back and the performance of the animal comes back to normal levels. What this suggests is that we've been thinking about memory and the hippocampal formation in entirely the wrong terms. And more generally, we've been thinking about the participation of the anterior thalamic nuclei in cognition generally also in the wrong terms. So in the figure that you have before you, what you have is a set of block diagrams of brain regions which participate in differing functions. And what you see is that the anterior thalamic nuclei participate in all three: memory, attention, and space. I haven't bothered to talk about our work on attention for reasons of time, but I have shown here that the spatial signal disappears after anterior thalamic damage, and learning, in other words, the representation of where you go in space is also dramatically affected after anterior thalamic lesions. So what does this mean? Well, this means that actually, as I've suggested at the start, that we've been thinking about how the brain represents memory in probably less than full terms. We've given a dramatic amount of emphasis to what we might call the temporal lobe memory stream, which is represented on the left here. And we've given relatively little attention to what we might call the diencephalic memory stream, which is represented on the right. When we look at the anatomy, what we see is that the inputs to these two regions are largely independent of each other, but the outputs of these two regions converge upon common cortical areas giving rise to the likely sites in the cortex for where memory consolidation is likely to occur. So let me conclude, because I don't want to run over time here, that what we have are distributed brain regions, the hippocampal formation temporal lobe on the one hand, and the anterior thalamic nuclei medial diencephalon on the other hand, which form a complementary partnership for episodic memory. And we know from looking at patients with brain damage that separate lesions to the hippocampal circuit or to the anterior thalamic circuit causes approximately equivalent deficits in memory. And we can replicate these deficits in experimental systems, such as in the rat or in the monkey. But we can also make a separate prediction, which is that if we focus on the anatomy in terms of the cortical sites of convergence between these streams, we will also cause memory impairments. And this resolves a really long-standing issue in the literature, which is that damage to particular cortical brain regions independently of the hippocampal formation can also cause amnesia. And our simple solution to this is to say that it arises out of the interaction between these two separate brain circuits converging in the hippocampal formation. I'd like to thank Bethany Frost, Sean Martin, Matthias Kefalkio, and Nurul Islam for their work in doing this, and also my longtime collaborator, John Aggleton, with whom we've published much of this work. The Wellcome Trust has supported this work over the years, and we're very grateful to them for their support. My social media coordinates are there on the bottom left if anybody's interested. I'm on Twitter, Instagram, and I also do a regular newsletter. Thank you very much. I will stop there.
Okay, so Shane, thank you very much for this very impressive tour de force through, I think, many, many years of research. And so I see a couple of people raising their hands, but let me just have two short questions as a start. So as this is not my field, I just wonder how many laboratories are out there who are able to reproduce these kind of things. So is it more the experience with all these complex lab technologies that is just a high basis, or is it the cleverness of actually doing the experiments?
So that's a great question. And when I started doing this kind of work about 20 years ago, I could name all of the laboratories easily. And I knew everybody. And now I still know the same people, but there are multiple laboratories that can do these kinds of recordings. There are many of them in Germany. There's a fantastic set of labs in Magdeburg, in Regensburg, in Munich, where you are, and many other places. But what you will find is almost everybody is recording in the hippocampal formation. It's a really easy structure to get at. It's placed dorsally in the brain in the rat, so it's easy to access. And a thing I've tried to do in my scientific career is keep away from areas that everybody else are working on, because there are lots of smarter people out there than me. So I might make some discoveries in some other part of the brain. So there are many labs. There are many in the UK. There are many in the US and Korea, Japan, and lots of other locations as well. France, the Berthoz and Wiener labs, and many others in Marseille and places like that. But typically people are recording in the hippocampal formation. And it's an area that physicists have contributed much to in terms of modeling. But it would be nice if physicists encouraged the neurophysiologists to move away from the hippocampus and think about other brain regions.
Okay, thank you. Now I've seen a number, so there was Jörn, I think you raised your hand. But he has disappeared. I just saw his hand. But there was someone else who had wanted to make a comment, but they all seem to have either disappeared or muted themselves. I don't know why.
I might make just a quick comment. There's a great opportunity for people interested in data science and modeling now because the age of big data has finally arrived in neurophysiology. There are a new generation of electrodes called neuropixels, which allow you to record from tens to hundreds of neurons simultaneously, either within or across brain regions. And the problem we're starting to have now is that there is too much data, and the analytic tools we have available are actually quite poor. They tend to revolve around the use of correlations and statistical tests like this. And it would be really great to see other approaches where you have from signal processing, I think particularly focused on these kinds of data sets because they're very, very rich, and trying to infer causal structure from them is a really significant problem. So anyway, it's just a general comment. If there's anybody there who's looking for a slight career shift and they have data science as an interest, there's a big need to do this.
It's good to know, and I think as well, the data can travel more easily than people. There may be even an opportunity to do this somehow by just sharing data. We have done a similar thing, and Jörn will talk about this, and some other talks will do this, where we had actually students working on muscle data and imaging, which goes in this direction. Now, I have my last question. So you talked about rats. I think you also mentioned monkeys, and you mentioned this one poor person with seizures who had this emergency operation and then had to suffer from the consequences. How far is one from actually doing systematic tests or experiments with humans?
So there's a vast literature. It's almost incomprehensibly large now. So there's many experiments on using fMRI in both regular participants and in patient populations. It turns out that there are what are known as agenesis patients. So these are patients who have brain regions that have not developed for one reason or another. So hippocampal agenesis patients are I won't say common, but they do turn up. And there are patients who have infections, particularly encephalitis patients, who have selective damage either to the hippocampal formation or to the anterior thalamus. And these patients show similar patterns to the same pattern as is shown originally by Korsakov and Lawson, but also in patient HM. So typically what is done now in patients who have epilepsy is the minimal level of surgery is performed on the hippocampal formation. Typically what is tried to be done is to restrict the surgery to a single hippocampal formation rather than two. And often a type of surgery known as a neocorticectomy is performed, where instead of lesioning the hippocampus itself, what you try and do is just remove some of the inputs to the hippocampus, and that tends to damp down the epilepsy as well. I should mention as well, there are also another category of patient who are neonates who have suffered febrile convulsions. So these are convulsions that arise from fever in the early years, and those convulsions are often accompanied by bilateral hippocampal damage, and those patients have memory deficits that are enduring as well.
Okay, so thanks a lot. So I think let's keep in touch, and if you could somehow think about some data that you could share easily with some questions, and then I could try in the next round of students to see if some student groups would be interested to actually tackle some of those questions that may lead to, well, either software packages or research grants or whatever. So it's very stimulating. And we have done this successfully, I think, with the collaboration with the DLR. And so that's a good opportunity to thank you again for joining again this year. And hopefully you're willing to join also next year.